when we have a colony on Mars, we want to make the medicines we need at Mars. So basically, we try to produce paracetamol using some algae in the Martian environment.
We are one of the few institutes in India that actually has an iGEM team.
We were lucky enough to bag a title sponsor, AstraZeneca.
Wow.
Most of our lab work and our competition fees comes from our corporate sponsorship.
Hi guys, welcome to Best Place to Build Students Edition. I'm Vidhi, a fifth year student in IIT Madras. Have you ever been curious about the term synthetic biology or genetic engineering? We're here today with Team iGEM of IIT Madras. Their team leads are here with us minutes before the orientation to explain us more about this. I'm here with the team lead from iGEM, Aldis, and we're here minutes before the orientation to know more. So, first of all, what is iGEM?
So iGEM is a competition that happens every year in Paris. So it stands for International Genetically Engineered Machine. So it's basically a synthetic biology competition where undergraduate and postgraduate teams from across the world come and solve real-world problems every year.
Okay, wow, that's a lot to take in at one time. So, first, what is synthetic biology?
All right. So yeah, synthetic biology is not that different from genetic engineering. So what we do is we apply genetic engineering principles from an engineer's mindset. So we consider bacteria as a system, and we want to make it produce something that we want, basically. So to do that, we introduced new genes and genetic circuits to make it do what we want it to do.
Okay, could you give me some example of this being used in the real world?
So one, I think, very widespread example would be insulin. So insulin is something that is normally produced in our own cells, and diabetes happens when you're not able to do it yourself. So what happens is scientists introduce the gene that produces insulin into E. coli, and then the cells then produce the insulin that we are able to then use, basically.
Just by introducing that, now your body is able to make use of that E. coli bacteria?
No, the
How does it-
bacteria is grown in a lab, the insulin is extracted, and then it's post-processed, and then we get it as medicine.
Oh, okay, so you're using a bacteria as like a manufacturing hub now for insulin
Yeah.
instead. That's really cool. And have you guys done any such projects like this in the past?
Oh yeah, actually, last year our project was based on space biomanufacturing. So what we did is, basically, transporting material in space is expensive. So when we have a colony on Mars, we want to make the medicines we need at Mars. So basically, we tried to produce paracetamol using some algae in the Martian environment.
Wow, that is really cool. So, how do you guys even come up with? Was this part of the iGEM problem prompt, or was this something that you guys were trying to innovate yourself?
No, so iGEM gives us complete freedom in terms of our problem statement design. So we take up new problem statement every year. So we do literature research. We reach out to different stakeholders in different domains of life, and we basically find some problem that is worthwhile, and then we figure out a way to solve it using synthetic biology.
Sounds really fascinating. And knowing your track record across the past years, I think you won like 3 or 4 gold medals consecutively.
Yeah.
I expect this year to like go the same or even better.
Thank you.
So I guess we're running out of time for your orientation, only about 5 minutes left. So can I know a bit more about the orientation itself? What's it for? What's happening today?
So my co-lead Karthik is right here, and he's the best guy to ask about this.
Sure, okay. Hi Karthik.
Hello, Vidhi.
Hi, thank you for taking the time. I know we're running short.
Yeah.
So what is this orientation about? The main purpose?
So the main purpose of this orientation is just to introduce freshers and even second year, third year undergrad students to the field of synthetic biology and help them understand what we do in iGEM and as well as the projects that are going on, so that the interested folks can sort of DM us and like get in touch with us and possibly join the team in the future.
And what sort of people typically come for the orientation or that you're looking for? Do they need a strong background in biology to even understand the orientation or what you guys will be doing?
So we're not looking for people who are well versed with biology and things like that. We're just looking for enthusiasm. And so these orientation sessions tend to happen when the freshies just join IITM and like get in touch with us. So This is sort of like just an introduction to what iGEM does, and I think Aldous previously mentioned of the synthetic biology and the kind of innovative the work that we do. And so this is just a way to like publicize our work to the newcomers.
Yeah, I didn't have much of an idea of it either if I wouldn't have been in IIT Madras and heard of iGEM and seen the
Right,
team.
right.
Yeah.
So iGEM as a whole is not just for biotech students. It also has a very interdisciplinary component to it. And with the dry lab projects and with the software stuff, with the HPs and things like that, we expect students from all departments to somehow contribute to our greater cause. And it also comes to the fact that iGEM teams tend to have a vertical and say web development, because the wiki that we design is a key component into our deliverables. And so we're just looking for people who have previous web development experience. And similar to that, we're also looking for like media team members. So yeah, all the freshers who have come here, they all might not be from the biotech department or have like biological knowledge, but if they know a bit of web ops or if they know a bit of media, they can always sort of contribute to our innovative ideas and solutions.
Wow. So it sounds like you guys have a lot of depth into whatever topic you're going, as well as like a lot of breadth
Right,
into the
right.
kind of roles that are there. And it's
Yeah, yeah.
like a really nice and very niche opportunity. Like I can't imagine any other place wherein you get to like learn about this or
For
even
sure,
have the
for sure.
facilities to build this.
For sure.
That's really,
So
really cool.
thank our departments and the other departments like the MST for offering their labs to, do such innovative work. I think the department is also slowly realizing that the world is headed towards biotech, and they need to sort of help us grow and like help the competition go and things like, because we come up with innovative solutions. Maybe one day we can publish a paper that's all that can be put into the Nobel that can be published in Nature, for example.
Typically, I've seen orientations just be like a presentation from the team and people listening, and it gets a little bit less engaging sometimes or a bit hard to comprehend. But in your mail, I saw you'd ask students to bring their laptops
Oh,
if
yeah.
possible. So, what's happening there?
Yeah, that's interesting because we expect people to learn immediately and quickly and even use AI tools to solve problems. So we would like put out some open-ended questions, and we hope that with like 10 seconds or 15 seconds in rapid fire, can students sort of pick up on it and give answers. And we hope that through some questions, we can judge how the enthusiasm of the freshers and then look for people who are interested in just the learning aspect.
Wow,
Yeah,
that
world is headed towards
yeah.
AI and like LLMs and things like that. So it's best that we know how to use these tools to help solve the problems that we're tackling.
Right. I mean, it's going to be a lot more engaging than other orientations.
For sure, for sure.
And you guys also get to gauge. Yeah. I'll let you get back to your orientation. Good. Best
Cool.
of luck
Thank
for
you,
it.
thank you so much.
We're here today at the biotech department in the lab space where iGEM works. We saw how they were recruiting people. Let's go see how they work. And how was the orientation?
Oh, the freshers orientations was amazing. We had over 50 people who actually showed up, and all of them were very enthusiastic about the work.
That's great to hear.
So you'll be rolling out apps soon, or does it happen?
Yeah, so that happens a little later. That happens in a month or two. For now, it's just to get them sort of introduced to the field of synthetic biology and keep them thinking as to what we're doing. And once the next year's project is decided and a lot of the groundwork is set, we can then roll out the applications.
Great. And is this like the main lab where you guys work, or do you work out of other spaces as
Yes,
well?
so this is one of the important labs. So we do most of the cell culture work here, but there are two other labs as well, which are run by mainly the professors who are in charge of our current project, Professor Mayappan and Professor Armopan.
Great. So I have a lot of questions to you. I'm left very confused after the orientation.
Yeah, I know. It's a bit overwhelming, yeah.
Let's get right to it.
For sure, for sure. I'll love to answer some of those questions, yeah.
So we're here where it actually happens and where you guys are in action. So what project are you guys working on this year?
Right. So 2025, our project revolves around what's known as gene regulations. Have you heard about gene regulations by any chance?
No, I had bio till 10th.
Okay.
Then I haven't seen.
Right. So essentially, you have DNA that converts to RNA and that converts to protein. What researchers is looking for is trying to overexpress these particular protein targets, so that way we can produce desired compounds or desired enzymes and things like that. So one way of doing that is by gene regulation. So you overexpress those gene targets. And this year, this is a very interesting field if you think about it. We have come up with a novel system using what's known as epigenetics. Have you heard about epigenetics before?
I've heard of genetics.
Genetics?
Epi is outer, but
Yeah, but very similar. So in genetics, you're sort of manipulating with the DNA strand, right? In epigenetics, what you're doing is adding methylation sites. So what a methyl group is a standard CH3. You would have studied it in 9th and 10th, right? So you add these methyl groups, or nature adds it, at different sites on the genome, and that way it sort of controls gene expression.
So instead of like you're not manipulating the DNA itself, but rather the outer part or
Exactly. So that's a good question that you asked, because normally gene regulation is controlled by, say, modifying the gene sequence itself. So you introduce what's known as different promoters. That way the entire mechanisms happens much quicker, and so you have more protein being produced. What we're doing this year is we're designing a novel system that involves what's known as a CRISPR-Cas system. So a Cas, I mean, it got the Nobel Prize actually. So it targets specific sequences in the genome, and what we are attaching to it is a DMT, which is a way for the cell to recognize, okay, these are the sites I want to mark for methylation. And so now you're able to methylate certain sequences, certain parts of the sequence, and so that way we are now allowing the natural process, that's the transcription factors, to produce more protein. So this is, I would say, a novel system that we are doing this year.
sounds really cool. But I'm not able to connect how does adding methyl groups on a DNA backbone affect how protein
Right.
is
So
produced?
one way it does it is by like modifying the secondary structure of the DNA. What you can think of is methyl groups acts as potential markers for transcription factors to go and bind. So the transcription factors, when they bind to the DNA sequence, they sort of produce more copies of the RNA, and then you have more copies of the protein. Now it has multiple applications. So say now you are able to mass produce an enzyme that produces an essential compound that's involved in a reaction that produces a metabolite of your interest. What that allows is you have engineered cells, which in itself is amazing. You have engineered cells that can now produce a compound that normally is found in very less abundance in now mass. And so that you could use that in various biomanufacturing plants and other
So you can have other drugs or like components or chemicals.
yes,
which are
so
rare to find.
because this is a system, now you can sort of engineer bacteria and add more genes, produce compounds, and then start overexpressing those genes, and now producing more enzymes, producing more metabolites. So that's essentially what we are sort of doing here. Yeah.
Wow. And how far along are you in this process?
So we are done making the system in the prokaryotes. So we've engineered E. coli to, produce this. Now we're just trying to see if we can show these results and show if we've successfully, modified the organism to produce what we intended to do. Yeah.
And so far, it seems like it's doing.
It seems like a
That is
yeah.
incredible. Wow.
Yeah. So we hope to present this during the actual competition, in the iGEM competition. We hope to get like a nomination, even win the best track prize for biomanufacturing and like therapeutics and things.
So, Karthik, I've been hearing all this fancy stuff that you guys do, like editing genes or putting DNA into bacteria. But how do you actually do this?
I mean, that's the big question, isn't it? We're doing all of this crazy stuff, and then, so why don't I just call one of my experimental members over here, Skanda? So he's been working on the project for over a year. He'll give you guys a better overview of how exactly we do this particular transformation.
Oh, hi, Skanda.
Hello, yeah, I'm Skanda. I'm part of the wet lab team here at iGEM, and I can show you, can give you an overview of a very important procedure that we do in every one of our projects, and which is very pivotal to the whole molecular biology aspect of work.
So, what process are you going to show me?
So this process is called bacterial transformation, which is essentially introducing new circular DNA into a bacteria. So let's say you want to make the bacteria produce a protein. So you use this procedure to introduce the gene for that protein in the bacteria.
Oh, so you just inject a gene to make the bacteria do what?
Not really. It is not as simple as it sounds. I can probably show you around
Yeah, sure.
So this is before we start, this is the laminar airflow This is basically an instrument which has air filters and UV lamps which make it very sterile for microbial works. Anytime we deal with bacteria, we do it inside the laminar airflow. First step in our procedure would be making the bacterial cells competent to take up the DNA. So bacterial cells usually don't readily take up DNA from outside. We treat bacteria with salt solutions at very low temperatures to make them competent to take up these circular DNA. And all this is done inside the laminar airflow. And once the bacterial cells are competent, we mix the DNA material, that is the circular DNA, and the DNA comes in tubes like this. This is called a microcentrifuge tubes, and we basically mix the bacterial culture and the DNA in this, and we incubate it in ice, and then 42 degrees for some time, and then back in ice. By this time, the bacterial cells would have taken up the circular DNA.
Okay, so basically this is a sterile hood wherein you're doing whatever manipulation with the bacteria and the DNA.
Right.
You make the bacteria healthy enough or like
Yeah.
ready enough
Ready,
to take
yeah.
up the DNA. You have the DNA ready, you mix it up.
Yeah.
After that, what?
And now the bacteria actually at this stage are under a lot of stress because this is not how they normally are, right? So we basically after some time we give them food. So food for bacteria is basically the broth. So this is
Oh.
this is called a an LB broth. This is basically a media in which bacteria thrive. So this is food and very conducive for their growth. So we add some of this LB broth into this tube. So bacteria kind of like they recoup and they revive, and we incubate this bacteria in something called an incubator. This incubator is an instrument which gives the perfect environment for the bacteria to grow. So it looks like this. It has temperature and pressure probes which
Okay.
continuously monitor all the conditions required for bacterial.
So there's some airflow conditions also which
Yeah, there are airflow conditions which are also maintained. Yeah, and after the bacteria have revived, that is,
how long do you usually keep them in there?
it is usually around an hour.
Oh, okay.
Yeah, and once the bacteria revived, you can actually see them grow. You can see the density, the optical density change. And once they are grown, we plate them. We spread this liquid culture on plates. So I'll just show you where the agar plates are kept. Yeah. So these are called agar plates. So this is basically a solid media for bacteria to grow on. Just like how the LB broth was a liquid media, this is a solid substrate on which bacteria grow. And so we spread this liquid culture on these plates, and we again incubate them in the incubator overnight. And after around 20 to 24 hours, they grow into something like this. So these are actually
Oh.
bacterial streaks.
So what are those red streaky kind of things I'm
Yeah,
seeing?
this is how. So that is the pattern in which we spread the bacteria after their revive. So they grow in something like this, and you can see single dots. They are called colonies. So all the bacteria in each colony are actually identical because each colony comes from a single bacterial cell. So yeah, we treat each colony as a single bacterial representation of a single bacterial cell. Yeah. Yeah, so now we have bacteria growing on agar plates, which actually have the plasmid taken up. So they have the circular piece of DNA inside them.
That was really cool. Okay.
Yeah, this is something we do on a very regular basis. This is very pivotal, very essential. It's a very essential part of any project that is done in like genetic engineering or molecular biology.
So do you guys have to work around odd times to be able to
Yeah, we do
make the
because
experiment
they
time?
we have specific time like time points where we need to check if the bacteria are growing or like take them out of some maybe transfer it from 80 degrees to 42 degrees or whatever. So yeah, we have to work at odd times, and Yeah, but it is pretty cool.
Yeah.
Yeah.
Thanks a lot. That cleared up a lot of things in my head, how it actually happens. I can
Yeah.
somewhat see it, not entirely.
No, it is fine. This is the basic overview of what is happening.
I have a couple of questions left about how the team functions on a day-to-day basis. What is their structure to execute and be at such a good stage already? So we have Aldis for that. So Aldis, yeah, as I mentioned, how do you guys have the team structured? Like I remember you mentioning that you're more of a computational guy, not in the wet lab much. So
So the form of a team really follows the function. So as you saw, we have the wet lab people who do the experimental work. We also have the computational team that works on modeling and the software development part of a project. So modeling is where we sort of computationally simulate the systems that we want to produce, like in experimentally, and see whether they'll work out or stuff like that. And in addition to that, we also have a web team. So they help us put out our work in a way that it's accessible to the public and who wants to see the work.
oh, okay. Yeah, I've seen your website and how it's like always updated and you can go through the different sections. That's really cool. And what about like other comp teams have sponsorship and
Yeah.
stuff for that? So is it funded through the professor itself or how does it work for you?
Okay. I think similar to most competition teams, most of our funding comes from corporate sponsorship. So all the members pitch in and we do outreach to multiple corporates. And this year, actually, we were lucky enough to bag a title sponsor, AstraZeneca.
Wow.
Most of our lab work and our competition fees comes from our corporate sponsorship.
Wow. And how big would your team be per se in each of these divisions?
Total, we have around 30 members in the team. So we have like 10 to 15 in each of Wet Lab and Dry Lab and a small 5-member WebOps team. And sponsorship and human practices also, all the members pitch in.
So you get to learn a lot in that way. Like you have the tech aspect and a bit of the management exposure if you would like to have it. That's great. And since this is very research heavy and you guys are working out of labs, I'm guessing you'll have some profs or PhD students helping you out?
Yeah.
Or-
So we have a couple of professors who guide us. So this year for our lab work, we are working with two cell lines. So one is a eukaryote, which is a mammalian cell line, and one prokaryote, as Kanta showed you. So for each of them, we are consulting with different profs who help us with the protocols and what to do. And of course, their PhD students also support us in the lab with the learning, helping us learn the skills that we need to.
iGEM doesn't seem like the typical competition team in the way it's structured and how it works. What do you think about that? Where does it sit in the whole spectrum of like tech teams in INSTI, competition teams or prof projects?
iGEM as a competition in itself is very open form, and it really encourages research and the mentality where you find a problem on your own and you devise a solution to it on your own and we implement it to fruition. So in that manner, it's a bit separated from other competition teams and clubs, and it's more close to a research project that one would take up in a lab.
So you have the research aspect while also having the whole competition drive and you
Yeah.
get to go there as well.
Yeah.
It's like the best of both worlds. Is what
Yeah,
I can see.
exactly. So this sort of exposure with the research work as well as the sort of management sponsorship and the outreach work is something that gives our members a very well-rounded exposure and makes them really valuable in whatever they want to do next.
So it sounds like a great experience. Are there any members in your team who are like thinking of continuing down in this field given their experience in iGEM?
Oh, a large majority of our team, of course, wants to go into biotech research and stuff like that. There are people who go into consult and stuff like that, and software development. So these are the major things. So I myself am a mechanical engineer. So where I look at myself going forward is there are research fields where really mechanical engineering and molecular biology come in together. So it's called an organ on a chip. So yeah, so for medical testing basically, so you are able to replicate those conditions in a chip basically.
Yeah, sounds fascinating. Organ on a chip has captivated many people's attention right now.
Definitely.
Yeah.
Definitely.
And is there any like next big technology that you're just waiting for or you think is going to shake the world?
Okay. I think the recent one was CRISPR, right? So that gives us this very selective way to change things however you want it to. And now, of course, I'd say the next steps in where the biggest frontier is in the computational side. So bringing in AI and because as you saw, each and every experiment is really difficult to do, right? So if you're able to take the burden off from that and onto computers, we can really speed up the research process, and the discoveries can be made so many more.
Like having like digital twins of everything, like a bacteria, even the circular DNA, how that mechanism would work out. So you can just simulate it
Yeah, yeah.
and then work it out. That would be really cool. Yeah. And how do you feel about like ethics and synthetic biology? I feel like there's such a fine line between innovating and even like just like you're manipulating life literally.
Yeah, yeah.
So there's a lot of responsibility in your hands.
Correct. So yeah, that's what. So when I said anything can happen, really anything can happen, right? So that's why large part of what we do is also responsibility and managing our impact and the ethics of whatever we do. So the most basic bioethics question is biosafety and contamination. So right now, whatever we do are contained in labs. So like I think Fanda might have told you about the antibiotic resistance. So if anything were to get outside the lab, it wouldn't be able to survive. So that basically ensures that we don't have an impact on the environment that we don't want it to. So that I think is the main ethics question. And then, so actually in all the jamborees that we present in, there's always a presence from intelligence agencies like the CIA and the FBI.
Okay.
Yeah, so they do awareness about bioterrorism threats and how we have to be responsible in the way that we conduct our work.
So what would you say is the main philosophy between like crossing the line with tech versus going towards a constructive path? Because I feel like, you know, technology has always just been a tool, like it's a knife, it depends on the hands in whom
Yeah,
it goes
yeah,
to.
true. So that's actually a very good analogy. So whenever you're working on something, you always have to consider how someone might use it nefariously. So if there's a possibility of what I'm working on being very, because it's just a sequence of DNA, right? You publish it, someone else can take it, modify it a bit, and say instead of helping save someone, it can just as easily be used to detriment to someone else. So that's something that we have to take responsibility of and make sure that, so if our work is going in such a direction, you stop there and then you just don't publish that work.
That was really nice chatting with all of you. Got to know a lot about iGEM and synthetic biology as such a fascinating field.
Oh, definitely. It was really a pleasure to talk to you and explain all this stuff and get some new perspectives in as well.